Hello class, and welcome back to our second video lecture here on Unit 5, the process of cellular respiration. The focus of this lecture video is going to be on the first process of respiration that we call glycolysis, and that process can be done by any type of cell that we know of that exists, either eukaryotic or prokaryotic. So let's dive right into it. So overall, there are three possible stages for cellular respiration. All right, the first being glycolysis that we're going to elaborate on this particular video. But then, as long as you can continue with the process of breaking down glucose, you can then move on to stage 2, which is called the Krebs cycle, otherwise known as the citric acid cycle. So something very similar to what we learned about in photosynthesis of the Calvin cycle. This particular stage cycle was discovered by Dr. Krebs. And sometimes it's also called the citric acid cycle because you're making citric acid. That's the first thing that is made that is a stable product in this cycle. And then finally, you can move on to stage 3, which is known as oxidative phosphorylation, which makes use of an ETC. Remember what that means, an electron transport chain. So this should look a little familiar, this term, right? So instead of photophosphorylation, where we use light to create a hydrogen ion gradient, that was then used by ATP synthase to make ATP. This time we're oxidizing something. And what are we oxidizing? Glucose. So over these three stages, what we're going to do is we're going to break down and oxidize glucose. We will use its electrons in an ETC to generate a hydrogen ion gradient that is also used to make ATP. So it's a very similar process, just the way we go about doing it will be slightly different. All right, but let's dive right into the first stage right now, which is glycolysis. All right, now, before we actually do it though, I just want you to keep this in the back of your head. The only reason why a cell would only do glycolysis and not move on potentially to the next two stages, 2 and 3, the citric acid cycle and oxidative phosphorylation, is if it didn't have one of these two things. Number one, oxygen must be present to do all three stages of cellular respiration. All right, oxygen is actually needed in the third and final stage of oxidative phosphorylation. If oxygen isn't present, nothing after glycolysis occurs. And secondly, do you have mitochondria present within the cell? So we're going to be focusing on eukaryotic cells mainly, all right? But glycolysis can be done in either prokaryotic or eukaryotic, all right? But if you're going to do all three stages, you also need to have mitochondria present, and only eukaryotic cells are going to have that, all right? Because stage 1, glycolysis, is going to happen in the cytoplasm to give you a little bit of ATP. But the real bulk of all the ATP that's produced from the breakdown of glucose will happen within the mitochondria in those next two stages, the citric acid cycle and oxidative phosphorylation. And you can see I have a much bigger, like, kaboom ATP image here, because that's where the majority of ATP can be made. For now, you're only going to make a little bit here in glycolysis, which is just fine for bacterial cells, or at least to get us started in eukaryotes. So, glycolysis can be done by either aerobic or anaerobic organisms. We learned about that in our last lecture, right? We have what's called obligate aerobes that need oxygen to break down glucose. We have obligate anaerobes that don't want oxygen to break down glucose. Then there was something else called a facultative aerobe, which didn't care. It could deal with oxygen, or it's fine if it doesn't have it. But glycolysis, it doesn't matter. Oxygen is not needed for this step. What we're doing is, all right, just in short, we're going to take one molecule of glucose, and we're going to break it in half. We're going to break it into two smaller 3-carbon molecules called pyruvate, sometimes also called pyruvic acid. This process will happen in the cytoplasm, which is why it can happen in either prokaryotic bacteria or eukaryotic cells, because all cells in existence that we learn from the cell theory have cytoplasm. And third and final, again, oxygen is not needed for this step to occur. So whether it's there or not, glycolysis will still occur. So let's go through the steps. I've broken it down into several steps for us to try to understand as the best we can. First step here, again, in the cytoplasm, we're going to have multiple enzymes. I'm not going to give you any names to memorize this time, but trust me, there's a lot of them that are going to add 2 phosphate groups to glucose, which will come from 2 ATP molecules and convert glucose into something new called fructose 1,6-bisphosphate. The bi here means 2. All right. In other words, what you did is you took a phosphate group, added it to the first carbon and the 6th carbon of glucose, which changes its shape into fructose 1,6-bisphosphate. But again, where did those 2 phosphate groups come from? ATP. So you have an initial input of energy here to get the whole process of cell respiration going, and that's going to be 2 ATP molecules. So I want you to think about that you're already negative 2 ATP in the whole. All right, but if you want to make money, what do you got to do? You got to spend money, right? So I just spent some money. This is my initial investment. What the idea is, I'm going to get more ATP back in the long run. Next, fructose 1,6-bisphosphate is split into 2 smaller 3-carbon sugars called, sounds familiar, right? G3P. So we're going backwards. So we just used 2 phosphate groups from 2 ATPs to turn glucose into fructose 1,6-bisphosphate, which then splits in half into 2 smaller G3Ps. And over here, you can kind of see that going on right now. I'll wait till it resets. There's fructose 1,6-bisphosphate. Right there, you have some bonds breaking, and you make 1 G3P. And you do make a second molecule down here that isn't G3P at first, but it does get converted into it. Now, G3P, though, in this process is not what we want, just like PGA wasn't what we wanted in the Calvin cycle. So our third step here is several more enzymes will take those 2 G3Ps, which have 3 carbons apiece, and turn them into what we really want, which are 2 3-carbon molecules called pyruvate, or another way of saying it is pyruvic acid. So this is the final product of glycolysis. When all said and done, besides the 2 pyruvates, you will walk away with 2 fully reduced electron carriers, and they're going to be NADH, not NADPH. And we would have made a gross total, I'll come back to that word gross in a moment, of 4 ATP molecules. All right, now let's see how it really was playing out. So bring your attention right here. Here's our glucose. We're going to add 1 phosphate group to the first carbon, a 2nd phosphate group to the 6th. These blue dots here would represent carbons like we were tracking in the Calvin cycle. And here's fructose 1,6-bisphosphate. All right, notice there's 2 phosphate groups. That came from our initial cost of 2 ATPs. Fructose 1,6-bisphosphate is split into 2 smaller G3Ps. You can see right here. And next, if we follow the arrows here, those 2 G3Ps, so right here, bring your attention, are going to get converted into 2 3-carbon pyruvates, or pyruvic acids. Now notice there are several steps going along to get there, right? From G3P to pyruvate, but I'm not going to have you memorize these little nitty-bitty, or nitpicky things, I should say. I just want you to remember that each G3P, as they're turned into pyruvates, will produce an NADH. So you're going to get 2 NADHs from the conversion of these 2 G3Ps, as well as 1, 2, 3, 4 ATP molecules total from the conversion of these 2 G3Ps into 2 pyruvates. So again, you all need to know this, but notice how there's another phosphate group coming in at this stage. To add that to G3P, you remove electrons that get added to NAD+, see how I got tripped up there, to make NADH. But this molecule here doesn't need those phosphates, so they're each going to be removed. So it's kind of weird, right? You add them only to remove them, but when you remove them, that helps you make 2 ATPs per conversion. That's actually called substrate level phosphorylation. I'll elaborate that more on the next slide. All right, so it's not oxidative or photophosphorylation. This part here in glycolysis is called substrate level phosphorylation. All right, now what that really means is this. You're simply using enzymes to transfer a phosphate group to ADP to make ATP. So right here where my laser pointer is, these two molecules which now have two phosphates on them, an enzyme is just going to remove one phosphate at a time, give it to one ADP to make ATP. It's a very slow and kind of tedious process. It's not really energy efficient. All right, but it's called substrate level because this molecule is acting as a substrate and this one enzyme is making one ATP. All right, so in conclusion, for glycolysis, our end result here is this. We're going to simply cut glucose in half. So we're going to take a 6-carbon molecule and turn it into 2 smaller 3-carbon molecules. That's really what glycolysis means again. Glyco is for sugar or glucose. Lysis means to split. It cost us 2 ATPs to get this whole ball rolling, so we're down a negative 2 ATPs. And total production, gross, is 4 ATPs and 2 NADHs, fully reduced electron carriers. Now, again, what do I mean by gross versus net? So those of you who have a job right now, you already know about this and how annoying it is. All right, you have a gross pay. So your paycheck will say, all right, you worked, however many hours you worked in a week, however many dollars per hour you were getting, multiply those together and you get your gross pay, which is before the dreaded taxes and other deductions. That's what you want to take home. But then you notice your paycheck is like almost 35 to 40% less. Well, that's your net pay. That's what you actually can walk away with after you remove taxes and any other wacky deductions that are coming out of your check. So this is your gross return. 4 ATP and 2 NADHs, but you really need to think of it as, what do you really walk away with after taxes? That's your net. So what are you walking away with? You're only really walking away with 2 ATP. Now, why is that? Well, you make 4 ATP gross, but don't forget about those 2 ATPs that you spent to get the whole ball rolling. Those are your taxes. So you got to subtract these 2 from the 4 that you made. So really what you're walking away with at this moment is only 2 ATP, which seems kind of useless, right? But the idea is we want to move further into steps 2 and 3 of cell respiration, where we can use these 2 NADHs, actually a lot more NADHs will produce, to create a gradient, a hydrogen ion gradient, that'll make a lot more ATP, as well as we can break down these additional 2,3 carbon pyruvates that we made, which will make us even more electron carriers, which will yield even more ATP during what we call, again, oxidative phosphorylation. So we cannot survive off of only 2 ATP per cell. All right, that's not going to cut it. This will only last you a couple minutes. Hold your breath right now. How long is it going to take you till you pass out? Probably only maybe 5 minutes if you could hold your breath that long, where your body would start breathing again and you'd be okay. But cells that don't have a mitochondria, this is all they can do. They can only do the process of glycolysis, and they're only ever going to get 2 ATP net from each glucose. I'm going to show you later down the line that we're going to potentially again get from 1 glucose, if we can do all 3 stages of cellular respiration, so we have both oxygen and mitochondria present, we can actually make upwards of 38 net ATP per glucose. Significantly higher than just 2, right? But for now, this is what all cells do to get the ball rolling. Glucose turned into 2 3-carbon pyruvates. You take away 2 NADHs. You did make a gross of 4 ATP, but don't forget that initial cost. Subtract those 2, and your net return of ATP is 2. If you have any questions, make sure you ask me during class. Thank you very much.